I am Risa Shinoda from Osaka University. Today, I'd like to talk about the AnimalClue, recognizing animals by their traces. We construct the AnimalClue dataset, which includes footprints, feces, eggs, bones, and feathers, and totaling 150,000 bounding boxes from 968 species and annotations for 22 ecological traits. Here is our motivation. So, identifying diverse animal species remains difficult as most models overlook nocturnal or camouflaged animals. While camera traps are widely used for wildlife monitoring, but they only capture animals when they pass directly in front of the camera. This means that even if animals are nearby, they won't be recorded unless they walk right in front of the sensor. While these citizen scientists often track animals by looking at the traces they leave behind. It's a simple but effective way to learn which species live in an area, even when the animals themselves are not visible. However, manual species inspection from traces requires highly domain-specific knowledge, which limits the scalability. And this creates a strong need for visual models that can identify species directly from such traces. Recent advances in computer vision have greatly contributed to automating key wildlife observation tasks, such as animal identification or activity recognition or pose estimation. However, identifying species from indirect evidence like footprints of fishes remain relatively underexplored, even though it plays an important role in wildlife monitoring. Here is some related work. So several previous studies have tackled trace recognition. However, their species coverage and the number of bounding boxes are limited, and most datasets focus only on classification tasks, which is not sufficient for realistic wildlife monitoring scenarios. In contrast, our AnimalClue dataset covers 5 major track types and provide a segmentation mask too. And also we annotate the 22 their species ecological traits. So here is the dataset creation part in our dataset. Firstly, we collected images and their species labels from iNaturalist, selecting only those that meet the both conditions. They are from research grade or they're from the Creative Commons license. After that, we use the grounding dino and segment anything to create initial masks. But however, sometimes we observe that there is unrelevant images in these initial masks. So we remove them. So that means that we remove the overlaid text images or animal in the frame, distant subject, or visible human facings. And also we modify some masks, and then we create the final dataset. This is an example of the final dataset. This is the statistics. So our dataset consists of 968 species, 200 families, and 68 orders. It includes a total of 159,000 bounding boxes across 5 dress types. This is the top 50 species distribution in your AnimalClue dataset with the vertical axis representing the log frequency. Let's move on to the experiment part. This is the classification benchmark. At first, we define the top 20% of species in our training split as the freaking categories, and the bottom 20% as rare categories in our training split. So overall, SwimB achieves the highest top accuracy among all baselines. FEDA shows the best accuracy across taxonomic levels, following by X, which also have the second largest number of species in our training split. This is a visualization of TSNE. As you can see, our fine-tuned clip models achieve the best feature separation, the right one, across all categories. And following this, BioClip perform better than the standard clip. But however, species level separation is still challenging, particularly for footprints and eggs. This suggested that the model should be trained specifically on trace data, even if they are pre-trained on large-scale animal image datasets. This is the detection part. RT et al. Achieves the best overall results, especially for frequent categories. However, detection of rare classes is still difficult, and also among trace types, feathers perform the best overall, maybe likely due to their large category diversity, and also distinguish their colors. Here are the segmentation results. Yellow V8, Yellow V11, and Mask Dino performs comparably across segmentation tasks. Among all trace types, here is also, feathers consistently achieve the highest MAP. Even though they include the largest number of species, this might be attributed to their distinguished color appearance. Here we show detection and segmentation results on AnimalClue using Yellow V11. Green box indicate correct predictions and red one are incorrect. We observe that the faint footprints or unclear traces often cause species misidentification, which reflects how challenging real-world wildlife data can be. And we also observe that the model misclassifies species that are taxonomically very close. The left one shows the model misunderstanding a southern lapwing egg as a masked lapwing egg, and the right one shows the model misinterpreting gravel scats as those of coyote. Here is the result for trait classification. We used swing transformer and evaluated by accuracy and F1 score. The left one is accuracy and the right one is F1 score. And the feather achieves the best results overall. Interestingly, for fishes, aquatic and predator traits showed highest accuracy, possibly because of clear morphological differences among species. These results suggest that trait influence depends on the trace types. This is our demo. So maybe user can put input images and then output should be a mask or bounding box and also their corresponding species names. Here is the summary. So we propose AnimalClue, a dataset for identifying animal species from their traces. This covers footprints, fishes, eggs, bones, and feathers. This totaling 159,000 bounding boxes from 968 species and annotations for 22 traits. We evaluate representative vision models and highest key challenges in trace-based identification. We hope this dataset advances research in animal tracking and wildlife conservation. Thank you for listening.